Hi, I'm Jeff Burns with the University of Kansas Alzheimer's Disease Research Center. I want to welcome you to the virtual data blitz for this year's Clinical Core Steering Committee. This year we are focusing on mitochondria and bioenergetics and Alzheimer's, why we should care. And this data blitz session accompanies the in-person or hybrid session and features 11 speakers who will present their data in short 4-minute talks. I want to thank the steering committee for choosing this important topic. And what we want to show is how much work is actually going on. So thanks to Howie, Cindy, Liana, Greg, and of course, Cerise, Nina, and Grayson for their support. So you're going to hear from these 11 speakers. Here's 5 of them and 6 more here. But you'll see from postdocs to professors, there's a whole lot going on in this area. So enjoy.
Hello, everybody. My name is Matthias Arnold, and I will be giving you a brutally brief metabolomics perspective on mitochondrial bioenergetics in AD. Before I do that, I want to thank the organizers for giving me the opportunity to present our work here. I also have to mention that all that I'm going to present is really the result of a huge team effort of many scientists brought together by Rima Kaddour-Daouk in the Alzheimer's Disease Metabolomics Consortium, as well as our partners in AMP-AD and associated consortia. Now, using the data we generated through AMP-AD and connected projects, we have compiled a large collection of metabolic changes observed in AD, with alterations of bioenergetic pathways being amongst the most prominent patterns we observed, as shown on the left. While in the periphery, we detected a complex disease stage-specific increase of bioenergetic dysregulation with accumulations of medium and long-chain acylcarnitines contrasted by depletion of other energy-carrying molecules like branched-chain amino acids. In brain tissue, we observed a global oversupply of molecules involved in bioenergetics that is somewhat reminiscent of blood profiles of individuals with type 2 diabetes. Now, when we compared our findings to the existing literature, we found that especially reports on metabolic changes in bioenergetic pathways were quite inconsistent. We therefore investigated modulation of metabolite associations with AD biomarkers across ATN measures through 2 AD risk factors, sex and ApoE4 status. On the right, we see results for A-beta pathology in the CSF, which revealed great heterogeneity of metabolic associations between stratified groups. Now, in order to examine if this heterogeneity could be used to perform data-driven substratification, we next applied unsupervised subgroup identification based on hierarchical clustering of fasting serum profiles of acylcarnitines, which are a central readout of mitochondrial energy metabolism. This identified several clinically different clusters consisting both of groups at more progressed disease states and a resilience cluster with individuals at old age, but less pronounced biomarker profiles. We then sought to disentangle genetic from potentially modifiable contributions to these bioenergetic subgroups. Genetic analyses revealed a number of significant associations with the clustering. However, this translated to only a few % of explained variance for the clustering structure. In contrast, acylcarnitine profiles cleansed from covariate effects, including genetic influences, explained a much larger portion of variance between the clusters, with a quite prominent pattern of higher contributions of markers for beta oxidation activity and efficacy. Now, despite these smaller genetic contributions, we wanted to investigate if we still could identify a particular pattern of non-modifiable risk linked to energy metabolism using multi-locus epistatic models. This revealed that genetic contributions seem to be limited to amino acid-based bioenergetics, a finding that we could replicate across ADNI, ROS-MAP, and the Mayo study of late onset AD. Now, combining these evidences, we defined the model of bioenergetic dysregulation in AD. We do know that brain glucose metabolism is deregulated early in the disease, which puts more demand on alternative routes of energy provision. Our findings suggest that the known age-dependent decrease in beta oxidation efficacy is accelerated in AD, and as soon as beta oxidation starts to fail, an unfavorable combination of genetic factors is detrimental, leading to global energetic dysfunction. As our findings indicate that the decrease in beta oxidation efficacy may at least in part be modifiable, we have now started to investigate this model in more detail. For this, we have derived a machine learning model that provides a composite metabolic score for mitochondrial function. We find that this score, which is based on peripheral metabolites, is strongly correlated to brain glucose metabolism measured by FDG PET. We further see that a high score is linked to later onset of cognitive decline, as well as a structural and cognitive reserve. Finally, longitudinal analyses suggest that genetic effects observed are indeed only relevant in individuals who will undergo cognitive decline in the future. These results are, of course, still preliminary, and we currently work on replication studies. However, if our findings should hold, they provide us with several exciting leads for potential therapeutic interventions. Thank you.
My name is Tony Grillo, and I am a postdoctorate fellow at the University of Washington and ADRC trainee in the Caberline Lab. And I'm excited to talk to you today about my work over the past few years, understanding how a genetic deficiency of mitochondrial complex 1 in the electron transport chain is able to induce tau aggregation, tau pathology, and that this tau pathology is dependent on the oxygen status. And so our group advocates there are shared mechanisms between mitochondrial dysfunction through complex 1 deficiencies in normative aging or age-related diseases, and in mitochondrial diseases such as Leigh syndrome. A leading model of Leigh syndrome are mice missing NDUS4, a subunit of complex 1 electron transport chain. These mice are short-lived, around 50 days of age, have very low activity of the electron transport chain, a neurometabolic phenotype, and are hyperoxic. And it was actually recently found that housing these mice at 11% oxygen instead of normoxic 21% conditions were able to extend lifespan from about 50 days of age for their normoxic NDUS4 knockout mice to greater than 500 days of age. And so I asked, what are the molecular mechanisms by which a complex 1 deficiency induces the brain degeneration and disease progression? To do this, I turned to the literature, and I observed there's actually significant similarities between pathology in mitochondrial diseases such as Leigh syndrome and in PSP, such as the brain regions that are affected. You see tau accumulation in the basal ganglia, cerebellum, brainstem for PSP cases. We observe brain lesions in those regions in Leigh syndrome, as well as complex 1 inhibitors, such as annonacin, were reported to induce tau accumulation. There's a cluster of cases in the Guadalupe Islands that was attributed to the local drinking tea from the soursop fruit containing this complex 1 inhibitor annonacin. And so I asked whether these NDUS4 knockout mice, which have complex 1 deficiency, show tau pathology, and whether this tau pathology is dependent or linked with the hyperoxic phenotype. And so I collected brains from these mice and ran Western blots against phosphorylated and total tau. And I observed the NDUS4 knockout mice that have been housed in 21% or non-moxic conditions showed about 5-fold increases in phosphorylated tau that was not observed when these mice were housed in low oxygen conditions. And this strongly suggests that the oxygen status and oxygen metabolism can influence the tau pathology caused by a complex 1 deficiency. Furthermore, teaming up with the Neuropathology Core and Dr. Dirk Keen at the University of Washington, I observed tau inclusion bodies in these knockout mice housed under normoxic conditions that was not observed in the hypoxic conditions. And so to better understand this, I teamed up and collaborated with Dr. Brian Kramer through the ADRC and used worms that he produced that are expressing human tau, specifically in the neurons. And I observed that intermittent hypoxia can extend lifespan in these tau transgenic worms. And so the tau transgenic worms generally live about 14 days of age, but intermittent hypoxia worms were able to live greater than 25 days of age. And this was able to reduce tau phosphorylation. And so I'd like to thank everyone in the group, the ADRC with Dr. Brian Kramer, Dr. Ilif, Dr. Keen, and others, and thank you for your time.
Hello, everyone. I'm Huihui Li, a postdoc in Ken Nakamura's lab at the Gladstone Institute. I'm very pleased to present my topic today. Neurons require glucose uptake and glycolysis. Brain is a highly active organ that relies almost entirely on glucose as its energy source. It's been widely hypothesized that neurons don't directly import glucose, or even if they do, they don't metabolize it through glycolysis. Instead, astrocytes export lactate to neurons, in effect bypassing glycolysis. Today, I will try to convince you that the neurons take up glucose and metabolize it through glycolysis to generate ATP. We generated 2 animal models targeting sequential steps in glucose metabolism. The first is GLUT3, which is the major glucose transporter in neurons. This model allows us to assess the impact of decreasing glucose import. The second is PCAM1, the primary neuronal isoform, which catalyzes the final step in glycolysis. Taken together, these 2 models allow us to assess the functional consequences of inhibiting glycolytic pathways in neurons. One of the behavioral tests that I'm showing is active place avoidance. Here we are looking at one aspect of the test, which is the number of times animals enter the aversive zone. Both GLUT3 knockout male and female show memory deficits start from 7 months old. The PCAM1 knockout female start show its deficits at 12 months old. This indicates that neurons need glucose and glycolysis to function normally. In addition to memory deficits, they also show sex-dependent changes in body weight. The GLUT3 knockout female gain weight slowly after 3 months old, and their hippocampal volume is much smaller, measured by T2 MRI scanner. These findings clearly indicate a sex-dependent difference in the requirement of glucose uptake, but we don't understand the basis for this yet. We used a thread-based glucose sensor to monitor glucose dynamics in individual neurons. The result shows GLUT3 knockout neurons have a much lower glucose baseline. Next, we performed the perfusion imaging on live neurons. As we can see here, in control neurons, give glucose, the level goes up, then we take away glucose, thread signal goes down. It stays down even further when we add in cytoclasm B, which is a glucose uptake inhibitor. In contrast, the GLUT3 knockout neurons are less responsive to external glucose change. We also checked if the decreased glucose can be translated into ATP. Here we used the ATP thread sensor. As is shown here, we didn't see significant ATP change at cell body, but we did see a remarkably decrease of ATP at synapses. This is consistent with our prior observation that synapses are particularly vulnerable to energy failures. To summarize what I discussed today, neurons directly import glucose and metabolize it through glycolysis. Our in vitro data further verified that neurons require glucose to maintain energy and function normally. Our further research will try to apply this framework to detect and understand the changes of glucose metabolism for Alzheimer's disease and targeted therapeutically. I want to acknowledge all the lab members and our collaborators who have done fantastic contributions to this project and our funding source. Thank you for your attention.
Hi, I'm excited to talk to you today about our work on muscle mitochondria in aging and MCI. So why mitochondria? Many proteins are metastable and have the propensity to aggregate under even mildly destabilizing conditions. So protective cellular responses, such as refolding these proteins, requires energy input, and bioenergetic failure can lead to protein aggregation. Now, the bulk of energy is produced in mitochondria, as we know. There are barriers to measuring mitochondrial function in the brain, but emerging evidence suggests that mitochondrial deficits could be systemic. To investigate this, we designed a 2-visit study with a GXT, blood draw, and muscle biopsy to characterize the whole body outcomes, including fitness and muscle mitochondrial metabolism. The full findings for this study have been published and can be accessed through the citation on this slide. Now, for this study, which we called ADMIT, we enrolled both cognitively healthy older adults and individuals with diagnosed mild cognitive impairment. Groups were comparable in terms of age, sex, and APOE4 carrier status. Now, we performed a muscle biopsy of the vastus lateralis muscle, and a portion was saved for RNA sequencing and protein analysis, We also saved additional tissue for immunohistochemistry. And finally, we immediately performed respiration analyses on fresh muscle samples for this study. We measured respiratory kinetics in the permeabilized fresh skeletal muscle fiber bundles under multiple substrate conditions. In the presence of the lipid palmitoylcarnitine, we discovered differences in ADP-stimulated respiration. We also performed additional titrations of substrates, including succinate and FCCP, and we saw consistent differences in oxygen flux throughout the experimental paradigm. Now, we also measured whole body fitness because we wondered if previous benefits we had observed in exercise studies could potentially be driven in part by changes in mitochondrial function. And as expected, we found a positive relationship between fitness and mitochondrial flux in cognitively healthy older adults and untreated MCI individuals. But in MCI individuals that had been treated with donepezil, we observed improved mitochondrial flux even at individuals with lower fitness levels. We also performed RNA sequencing, and this revealed 138 differentially regulated genes between control individuals and MCI participants. Gene ontology analyses indicated that biological processes involved in steroid biosynthesis, muscle metabolism, and lipoprotein assembly were significantly enriched. The differences between the MCI and the NCI individuals treated with donepezil were particularly robust, with nearly 700 differentially regulated genes. Network analysis revealed fatty acid metabolism, protein lipid catabolic processes, and protein biosynthetic process terms to be enriched. Now, finally, given our findings related to mitochondrial respiration, we also performed a focused analysis on mitochondrial genes. We found similar patterns of downregulation in MCI that appear to be recovered in donepezil-treated individuals when examining a number of key genes, including succinate dehydrogenase and the largest subunit of complex 1, as well as proteins involved in mitochondrial fusion, biogenesis, and survival-related proteins. So in conclusion, and thinking towards next step, we really found that mitochondrial function and gene expression are changed in MCI compared to cognitively healthy older adults. And medication use is an important consideration when examining mitochondrial function in cognitively impaired individuals. And a commonly used medication, donepezil, might have effects on other parts of the body that we didn't really acknowledge at this point, such as mitochondria. Mitochondrial function tracks with cardiorespiratory fitness in individuals who were not treated with donepezil, and additional work is needed to elucidate the effect of E4 on some of these mitochondrial outcomes due to our small sample size. So that was really brief, but I'd like to just thank my laboratory as well as collaborators. John Tifo was a key collaborator on this study as well as his lab. Our funding, this was funded by NR21 from the NIH, and of course, our philanthropic donors and our research volunteers who, without this, this never would have been possible. Thank you.
Thank you for the invitation to present our work on the association between mitochondrial DNA copy number and dementia and the phenotypes in TOPMed. This work is led by Yankei Zhang and with contributions from several investigators. Mitochondria are the powerhouse of most human cells, and mitochondrial DNA copy number is indicative of the energy generating capacity and metabolic status in a given tissue. The brain is very energy demanding, and neurons contain several tens of thousands of copies of mitochondrial DNA. Our previous work has shown that copy number declines steeply after age 60, and this decline is related to increased obesity, hypertension, hyperlipidemia, and diabetes. However, although previous studies have related mitochondrial DNA copy number to neurological disorders, we know very little on the association between copy number and dementia and the phenotypes. Therefore, we set to investigate the association of mitochondrial DNA copy number with cognition and MRI markers in diverse community-based cohorts from TOPMed. Our sample included over 19,000 participants from 9 cohorts, representing 57% women and a mean age of 59. Mitochondrial DNA copy number was derived from whole genome sequencing and was calculated as twice the ratio of the average coverage of mitochondrial to nuclear DNA. Because data collection in cohorts vary, we looked at 2 time windows of association. First, cross-sectional analysis, when the time between blood draw used for whole genome sequencing and collection of neurological assessments were performed in a window of 5 years. And second, a prospective analysis in which neurological outcomes were assessed 5 to 20 years after blood draw. We used linear regression models to assess the association between standardized mitochondrial DNA copy number and either MRI markers, including total brain, hippocampal volume, wide-body hyperintensities, or general cognitive function derived from at least 3 cognitive tests assessing distinct cognitive domains. Our primary models were adjusted for age, sex, time difference between assessments, cohort-specific covariates if needed, and education when modeling cognitive function. Additional models adjusted for cell blood counts and cardiometabolic traits. And finally, we performed inverse variance meta-analysis with fixed effects. We observed a significant association between higher mitochondrial DNA copy number and better general cognitive function, both within 5 years of blood draw, as well as 5 to 20 years after. These results remain significant after additional adjustments for cardiometabolic traits and cell counts. We did not find associations between mitochondrial DNA copy number and MRI markers. To conclude, our results suggest that higher mitochondrial DNA copy number is associated with current and future general cognitive function. However, our findings do not indicate such an association for MRI markers, but these analyses were limited in sample size, and the meta-analysis showed high heterogeneity. Our group has begun to assess the association between copy number, enteroplasmic mutations, and the incidence of dementia. We thank our sponsors, the NIA and NHLBI, for supporting this investigation. Thank you so much for your attention.
Hi, my name is Wolfdieter Springer. I'm an associate professor of neuroscience at Mayo Clinic in Florida. The pink-Parkin ubiquitin axis is the best characterized mitophagy pathway that identifies, labels, and clears damaged mitochondria via the autophagy lysosome system. A loss of either gene is linked to early onset Parkinson's disease. Accumulating evidence suggests a much broader involvement in aging and across neurodegeneration, as it is thought that failure to properly dispose of dysfunctional mitochondria will lead to the buildup of damage and over time result in neuronal death. The pink-Parkin pathway is dynamic and typically operates at very low levels to continuously survey health and function of mitochondria. The ubiquitin kinase pink accumulates locally at sites of mitochondrial damage and recruits and activates the ubiquitin ligase Parkin from the cytosol. Both enzymes together then decorate damaged mitochondria with phosphorylated ubiquitin that acts as a specific mitophagy tag, labeling damaged mitochondria for their degradation. Over the years, we have developed and carefully characterized antibodies against phosphorylated ubiquitin, which have really allowed us to demonstrate the pathophysiological relevance of the pathway. We've used these antibodies to follow and quantify the mitophagy tag in cells and in neurons, in vivo in mice, as well as in human autopsy brain, and more recently also in clinical samples. Aside from the expression and activity of pink and Parkin that produce phosphorylated ubiquitin, the levels of the mitophagy tag are determined by the amount of mitochondrial damage and the degradation rate of the autophagy lysosome system. In human autopsy brain, we found phosphoubiquitin levels to increase with age and independently with alpha-synuclein and tau aggregation in different neurodegenerative diseases. While we have thus far determined the individual contributions, we're currently expanding to other diseases to study combined and potentially synergistic effects of different comorbid pathologies, as well as the respective underlying mechanisms that may differ from disease to disease. Beyond these studies, we wanted to determine whether we can use phosphoubiquitin levels as a quantitative trait to identify novel genetic modifiers in an autopsy brain genome-wide association study. We identified brains with available genetic information and stained phosphoubiquitin on hippocampal sections from about 1,000 Lewy body disease cases. The sections were then automatically scanned and quantified to determine the levels of the mitophagy tag. Preliminary analysis resulted in the identification of the ApoE4 risk allele as the most strongly associated hit, reaching genome-wide significance levels. In addition, we identified several other loci that were suggestive but are associated with either particularly low or high levels of phosphoubiquitin, and we're currently in the process of replicating and validating those one by one. We further set out to explore whether phosphoubiquitin could be clinically useful as a quantitative biomarker of mitochondrial damage. Capitalizing on a recently developed ELISA, we analyzed plasma samples from age and sex-matched cohorts of neurologically normal controls, mildly cognitively impaired individuals, or patients with Alzheimer's disease, and found small but significant increases of phosphoubiquitin in both MCI and AD groups. We're now in the process of comparing our results with the more established amyloid tau and neurodegeneration markers and perform more detailed clinical pathological correlations. Similar to human autopsy brain, we do see both age and disease-dependent increases of phosphoubiquitin also in patient blood plasma. To summarize, the pink Parkin pathway seems broadly neuroprotective, and beyond its clear link to early onset Parkinson's disease, mitophagy defects may well contribute to other neurodegenerative diseases. Phosphorylated ubiquitin, the mitophagy tag, can be used as a specific and quantitative marker of mitochondrial damage. Its levels are not only dependent on pink and Parkin, but also on the amount of mitochondrial damage and the flux and degradative capacity of the lysosome system. Alterations in mitophagy seem to be prominent early features in both clinical and pathological samples from various neurodegenerative diseases. Despite similar outcomes, the underlying mechanisms for the observed increase in phosphoubiquitin may differ from disease to disease. With this, I want to thank the individuals in my lab, collaborators, funding agencies, and for your attention. Thank you very much.
My name is Andrea Stojakovic. I'm a scientist at Dr. Rosalind Anderson Laboratory at the University of Wisconsin-Madison. Today, I would like to present you with the work that is currently ongoing in the lab, as well as with my published work at the Mayo Clinic. The topic of my talk today is mitochondria targeted therapeutics for Alzheimer's disease. Currently, we're trying to understand the effect of caloric restriction on glucoregulatory mechanism in the brain of rhesus macaque monkeys. For this particular study, a cohort of male and female rhesus macaque monkeys were recruited to the study at 8 years of age, and they were randomized into control and 30% caloric restriction diet group. At 28 years of age, FDG PET scan imaging was conducted, and we found that caloric restriction significantly increased the glucose uptake in several brain regions, such as temporal lobe, hippocampus, parietal lobe, somatosensory cortex, and dorsal striatum. We further on wanted to understand how glucose uptake in the brain correlates with the insulin signaling pathway. For that reason, we did immunohistological quantification of insulin receptor and AKT in denti gyrus. And we found that level of insulin receptor was significantly increased with caloric restriction in both males and females, in particular in granular and molecular layer of denti gyrus, while AKT was significantly increased with caloric restriction in both males and females in all 3 layers of denti gyrus. Thereon, we conducted the fluorescent lifetime imaging microscopy to quantify the total levels of NAD and NADP. We found that caloric restriction significantly increased levels of NAD and NADP in granular and molecular layer of denti gyrus, suggesting improved mitochondrial metabolism. In my previous lab in Mayo Clinic, we conducted a study where we wanted to understand the therapeutic effect of mitochondrial complex 1 inhibitor, namely CP2, in a cohort of female APPPS1 mice. For this study, non-transgenic and APPPS1 mice were set on a vehicle or CP2 treatment at 9 months to the 23 months of age. At around 18 months of age, we conducted a Morris-Water maze and null object recognition test, and we found that CP2 significantly increased the memory performance in APPPS1 mice. We also did FDG PET scan imaging, and we found that, again, CP2 significantly increased the glucose uptake in the brain. We conducted also electrophysiological recording of long-term potentiation in hippocampal slices of these mice, and we found that CP2 significantly increased LTP in both non-transgenic and APPPS1 mice. We also did 3D EM imaging of dendritic spines in CA1 region of hippocampus and found that CP2 treatment significantly increased the number of mature dendritic spines. We also did similar study in 3XTG mouse model where both males and females were recruited to the study at 3 months of age, all the way to the 18 months of age. We found in Morse water maze that both males and females on the CP2 treatment had improved cognitive performance. FDG PET scan showed that both males and females have increased glucose uptake in the brain. And in females, by electrophysiological recording, we found that LTP was significantly improved. In all, we could say that changes in the brain metabolism that occur naturally with age create vulnerability for a range of diseases such as AD. Moreover, we could say that pharmacological or non-pharmacological interventions targeting specifically mitochondrial metabolism could activate several signaling pathways that are involved in the cellular stress responses, which are beneficial not only for the neuroprotection, but also ameliorating the pathological phenotype of AD. Lastly, I would like to give big thanks to my current lab, Dr. Barbara Brandlin lab, and my previous lab at the Mayo Clinic. Thank you.
My name is Eugenia Trushina. I'm professor of neurology and pharmacology at the Mayo Clinic, where I direct mitochondria, neurobiology, and therapeutics laboratory. The work in my lab is focused on the understanding of the mechanisms of mitochondria function and dysfunction in neurodegenerative disorders, including Alzheimer's disease. We use advanced techniques to study mitochondria dynamics, function, bioenergetics in animal and cellular models, including human iPS-derived brain cells. We establish small molecule mitochondria targeted therapeutics and use systems biology approach to establish translational biomarkers of efficacy. Mitochondria respond to stress by activating multiple mechanisms to ensure intact energy homeostasis. This is very important for cells with high energy demand, such as neurons. We identify small molecules that penetrate the blood-brain barrier, accumulate in mitochondria, where they partially but specifically inhibit the activity of complex 1. Such inhibition of the electron transport chain activity induces mild energetic stress and multifaceted adaptive mitochondrial signaling. Treatment with our tool compound CP2 in pre and symptomatic mouse models of Alzheimer's disease was found efficacious in restoring synaptic function, reducing inflammation and oxidative stress, levels of A-beta, phospho-tau, ultimately blocking the ongoing neurodegeneration. This treatment was also found beneficial, extending health and lifespan in chronologically aged mice and mice fed with a high-fat diet. Treatment was more efficacious in female mice. Multiple studies included evaluation of efficacy using electrophysiology, histochemistry, specifically looking at inflammation, levels of A-beta aggregation, neurodegeneration in locus coeruleus, which mimics situation in Alzheimer's patients. We also look at dendritic spine morphology using advanced three-dimensional mitochondrial electron microscopy reconstruction, demonstrating efficacy of compounds where mechanisms converged on the activation of MPK. Translational validation of the efficacy was done by cross-validating mouse RNA sequencing data with human RNA data available from the AMP-AD knowledge portal. Data suggest that treatment reversed major AD mechanisms, including inflammation and synaptic dysfunction. Therapeutic efficacy of our compounds in vivo could be monitored using translational biomarkers FDG-PET, phospho-NMR, and blood-based biomarkers. Clinical indication for the development of small molecule partial complex 1 inhibitors include slowing down cognitive decline and improvement of cognitive functioning in patients with mild to moderate cognitive impairment due to Alzheimer's disease. We aim to develop oral tablet or capsule for chronic administration in aging population. Current stage of development of the drug discovery program includes 3 US patents that were granted and the support from multiple agencies, including Alzheimer's Drug Discovery Foundation and Huntington Discovery Institute. We were supported by these 2 funding agencies primarily from 2009 to 2019. And we are in the second year of the now NIA-funded grant through Blueprint Neurotherapeutics Network Program, with the ultimate goal to develop clinical candidate for phase 1 clinical trial in 2026. We have multiple publications in support of the premise. We have multiple collaborators on these publications who independently validated efficacy of our compounds in their laboratories. This work was supported by multiple collaborations, multiple funding sources, and talented individuals working on this project in my lab. Thank you very much for your attention.
Hi, my name is Wangjia Wang. I appreciate the opportunity to present my work. Today, I'm going to discuss a less known aspect of mitochondria, which is their potential role in mediating cellular microRNA activities, especially for a subset of inflammatory responsive microRNAs. This work is supported by the Kentucky Spinal Cord and Head Injury Research Trust, and I thank my colleagues for their tremendous contributions to this work. MicroRNAs are a family of small non-coding RNAs. The mature microRNAs are single-stranded of 21 to 24 nucleotides. MicroRNAs regulate gene expression post-transcriptionally. Therefore, the regulation can be local and rapid, which is critical in CNS gene regulation. MicroRNAs are important regulators of all normal biological and pathological processes. This regulation of microRNAs are associated with almost all human disease and pathological conditions, including neurodegenerative disease. Previously, microRNAs were thought to function in cytosol only, but recent studies reveal that they are also localized and function in several subcellular organelles, such as nucleus, ER, and mitochondria. Initially, when we isolate rat brain mitochondria for microRNA profiling, we found several inflammatory responsive microRNAs, including microRNA 142.3p, 142.5p, microRNA 146A, and microRNA 150. These microRNAs are significantly enriched in mitochondrial fractions relative to cytosols. Our latest studies show that these same inflammatory microRNAs are also enriched in mitochondrial fractions isolated from human brain tissue. These experiments demonstrated a conserved phenomenon and perhaps biological function in both rodent and human brain tissues. Interestingly, these same subsets of inflammatory microRNAs are found to also enriched in the junction between mitochondria and endoplasmic reticulum, called mitochondria-associated ER membrane, or MAMs. Relative to ER fractions, these microRNAs are enriched in both mitochondria and MAMs. When we test whether mitochondrial stress will affect the microRNA activity using uncoupling agent, DMP, it showed that mitochondrial uncoupling resulted in a subcellular compartmental shift from mitochondria to MAM. When subjects rest to traumatic brain injury, we observed that the levels of these inflammatory microRNAs reduced in MAMs and increased in ER. These preliminary studies reveal that a subset of inflammatory microRNAs are specifically associated with mitochondria and MAMs in brain. Mitochondrial dysfunction affects the activity and function of these inflammatory microRNAs, and we hypothesize that Mitochondria play a role in mediating inflammatory microRNA activities via mitochondria MAM-ER network. Thank you.
Thanks for the opportunity to share my recent work. My name is Wenzhong Wang from the Case Western Reserve University. The title of my talk is ApoE4 mediates abnormal mitochondria in the lysosome context in Alzheimer's disease. AD risk ApoE4 involved in many aspects of AD pathogenesis. As shown here, ApoE-associated lipoparticles transport a beta oligomers into neuronal cytoplasm by specific ApoE receptors that accumulate a beta and ApoE in the lysosome system, damage the integrity of the lysosome system. In the meantime, mitochondrial dysfunction is also well characterized in AD neurons, as shown on the left of the slides, and we can appreciate the fragmented mitochondria. Therefore, it is still unknown whether there is any connection between two pathological features in AD neurons by ApoE4. We hypothesize that the abnormal crosstalk between mitochondria and the lysosome might cause the buildup of mitochondria beta-42. We first analyzed the crosstalk between two organelles by electron microscopy analysis in human AD cortex. In the panel A, we could define two types of endocytic vesicles. We found type 1 vesicles with the electron dense contents and type 2 with the light content in the lumen of vesicles. Quantification suggests that increased association of both types of vesicles with the mitochondria in AD samples. In the panel B, immunofluorescence analysis with the mitochondrial marker and in lysosome markers suggests that ApoE4 was associated with increased mitochondria and in the lysosome context in AD neurons. To explore the underlying mechanism, we carried out an unbiased screen of ApoE4 protein interactome in neuronal cultures by co-immunoprecipitation assay with anti-ApoE antibody. In panel A, pathway analysis of ApoE4 interactome suggests the involvement of endocytic vesicles and late anisone. Among the top-ranking interacting proteins, we focus on the lysosome memory protein study 3, which is essential for transport mitochondrial cholesterol. We confirmed that co-immunoprecipitation of ApoE and study 3 protein with either anti-ApoE or study 3 antibody in panel B, suggesting the robust co-immunoprecipitation of both proteins. We then ask whether study 3 affects mitochondria in lysosome context. We will all express or knock down study 3 expression in neurons and analyze the mito in the lysosome context by proximity ligation assay with the mitochondria and lysosome markers in panel A, and show that study 3 all expression increase the mito and in the lysosome context. By contrast, knock down study 3 abolish such context in neurons. In the meantime, we measure the mitochondria beta levels in the right panel. All expression study 3 increase beta levels in mitochondria, but knock down reduce beta level in mitochondria. In conclusion, our data suggest that study 3 is a likely downstream player of ApoE to transfer beta into mitochondria. In a short summary, our study proposed that in the cytosol, A-beta and ApoE4 promoted the study 3 dependent mitochondria in lysosome context, by which way A-beta was translocated into mitochondria to induce mitochondrial dysfunction and neuronal loss in AD. Finally, I would like to give my thanks to their members and funding support of this study. Thanks for your attention.
Hi, everyone. Today I'm going to talk about how mitochondrial dysfunction regulates ApoE expression and secretion. And this is work that's been funded in part by the ADRC at Emory. So to start out, I want to talk about a couple of models for how mitochondria are able to influence Alzheimer's processes. So in a more typical model, mitochondria are downstream from A-beta plaques and the ApoE4 allele. ApoE4, of course, being the strongest genetic risk factor for late onset Alzheimer's disease. It's been shown that both plaques and the ApoE4 allele lessen respiratory gene activity, and so in this way, mitochondria are just acting kind of in response to plaques and ApoE4, and they're downstream. An alternative model is one where mitochondria are able to influence ApoE and A beta plaques. And in this model, mitochondria would be part of a feed-forward loop that's contributing to disease processes. And so this is the model that we favor based on the data that I'll show you. And a prediction of this model is that genetic or pharmacological disruption of mitochondria would affect ApoE expression. We'll be focusing on this link between ApoE and mitochondria. And to test this hypothesis, we targeted complex 3 of the electron transport chain with antimycin in iPSC-derived neurons and astrocytes. And we did this with an ELISA-based assay. So if you look here, in gray is the vehicle condition, in blue is the antimycin treatment. For the neuronal lysates, we saw no change in ApoE levels. And looking at astrocyte lysates, we saw about a 2-fold increase in ApoE levels. And then in conditioned media from astrocytes, we saw a similar about 2-fold increase in ApoE levels. And so we were able to look at the ApoE that's secreted into the media over these 48 hours of antimycin treatment. And we looked also at the mRNA level at ApoE levels for first here the astrocytes, where again in gray we have the vehicle treatment, and then in blue here we have the antimycin treatment, and we saw a significant increase. And this was done using nanostring, which is an mRNA quantification that doesn't use any amplification step. And we looked also at a couple of genetic mutant mitochondrial lines, one lacking the protein SLC25A1, which is a carrier that moves the metabolite citrate on the inner membrane, and then a second line, which was lacking the ATP translocator on the inner membrane. And when we look at both these cell lines, we see a very robust ApoE upregulation. And this is just a loading control showing these different samples were loaded about evenly. Looking more at the results from this nanostring panel, which was an 800 gene panel looking at neuroinflammation. These are the volcano plots where for both the cell lines we tested, you can see the yellow are the upregulated hits. We see ApoE here, as I highlighted previously, and then the same is true in the antimycin treated astrocytes, where we see ApoE is an upregulated hit. And up top here is the proportion of the panel that was a significant hit. So comparing the cell lines to the astrocytes were had more hits here. But regardless of the number of hits, when we look at the gene ontology terms that are related to the upregulated hits in both cell lines and the antimycin-treated astrocytes, we see inflammation popping out, inflammatory response, cytokine signaling, cytokine receptors. And so this suggests that the mechanism linking ApoE and mitochondria could be inflammatory signaling, although we need to follow up on that. And this also shows that mitochondria are able to drive neuroinflammation. And so to sum up, I've shown you that inhibiting complex 3 of the transport chain is able to cause ApoE upregulation, that this ApoE upregulation co-occurs with increased inflammatory pathway signaling, and that mitochondria could act upstream in Alzheimer's disease through modulating ApoE-dependent disease processes. So this is a way that mitochondria can influence Alzheimer's outside of just bioenergetics. And we think it would be interesting to look more into alternative ways that mitochondria can influence disease outside of just bioenergetics. And I do want to acknowledge my lab and our collaborators, the core facilities that helped us, and my funding sources, including the ADRC at Emory. And we're about to release this work as a preprint, so please check that out if you're interested. And thank you for your time.
Well, that wraps it up. I hope you enjoyed that. I hope you can see how much is going on in the field of bioenergetics and mitochondria focused on Alzheimer's disease. And we see a bright future for this with a lot more studies to come. And we hope we did a little something to help set the stage for that. So thank you for joining us. Appreciate you tuning in.